Hi dear students. So in the last session, we concluded with the positive regulators of cell cycle. Now let's see the negative regulators of the cell cycle. One of the main negative regulator is the RB protein. While it is hypophosphorylated, it holds the cycle in check. Inhibitors of the CDKs also serve as negative regulators and their main action being at checkpoint 1. There are two families of inhibitors. The CIP family, that is the CDK inhibitory proteins. It is also termed KIP or kinase inhibitory proteins. It includes the proteins P21, P27 and P57. The second one is the INK family, that is inhibitors of kinases. It includes the proteins P16, P19 and P15. The action of P21 serve as an example of the role of a cyclin and CDK inhibitor. This protein P21 is under the control of the P53 gene, which is a particularly important negative regulator which is relevant in carcinogenesis, and it operates at checkpoint 1. The CDK cyclins and the APC are direct regulators of cell cycle transition, but they aren't always in the direct control. Instead, they respond to cues from inside and outside the cell. These cues influence activity of the core regulators to determine whether the cell moves forward in the cell cycle. Positive cues like growth factors typically increase activity of CDKs and cyclins, while negative ones like DNA damage typically decrease or block the activity. As an example, let's examine how DNA damage halts the cell cycle in G1. DNA damage can and will happen in many cells of the body during a person's lifetime, for example, due to the exposure to the UV rays from the sun. Cells must be able to deal with this damage, fixing it if possible and preventing cell division if not. Key to the DNA damage response is a protein called P53. It's a famous tumor suppressor and is often described as the guardian of the genome. P53 work on multiple levels to ensure that cells do not pass on their damaged DNA through cell division. First, it stops the cell cycle at the G1 checkpoint by triggering the production of CDK inhibitor proteins. The CKI proteins binds to CDK cyclin complexes and block their activity, buying time for DNA repair, and the P53's second job is to activate the DNA repair enzymes. If DNA damage is not fixable, P53 will play its third and final role, that is triggering programmed cell death so that damaged DNA is not passed on. In this diagram you can see that when a DNA damage occurs, the P53 will stop the cell cycle at the G1 checkpoint and will trigger the production of CDK inhibitor proteins, and these CK proteins will that is CDK inhibitor proteins will bind to CDK cyclin complexes and block their activity and thereby pauses, and the cell will pause in G1, and in the meantime the P53 will be trying to activate the DNA repair enzymes. By ensuring that cells don't divide when their DNA is damaged, P53 prevents mutations from being passed on to daughter cells. When P53 is defective or missing, mutations can accumulate quickly, potentially leading to cancer. Indeed, out of all the entire human genome, P53 is the single gene most often mutated in cancers. P53 and cell cycle regulations are key topics of study for researchers working on new treatments for cancer. Now let's see the characteristics of cancer cells that distinguish it from normal cells. The first one is that the uncontrolled proliferation, then the dedifferentiation and loss of function, invasiveness and metastasis, that is the tendency to spread to the normal functioning cells. In general, mutations of two types of cell cycle regulators may promote the development of cancer. That is, the positive regulators may be overactivated and it becomes oncogenic, while the negative regulators called tumor suppressors may be inactivated. So what are oncogenes? Positive cell cycle regulators may be overactive in cancer. For instance, a growth factor receptor may send signals even when growth factors are not there, or a cycle may be expressed at abnormally high levels. The overactive forms of these genes are called oncogenes, while the normal, not yet mutated forms are called proto-oncogenes. This naming system reflects that a normal proto-oncogene can turn into an oncogene if it mutates in a way that increases its activity. Mutations that turn proto-oncogenes into oncogenes can take different forms. Some change the amino acid sequence of the protein, altering its shape and trapping it in an always-on state. Others involve amplification, in which a cell gains extra copies of a gene and thus starts making too much protein. In other cases, an error in DNA repair may attach a proto-oncogene to part of a different gene, producing a combo protein with unregulated activity. Many of the proteins that transmit growth factor signals are encoded by proto-oncogenes. Normally, these proteins drive cell cycle progression only when growth factors are available. If one of the proteins becomes overactive due to mutation, it may transmit signals even when no growth factors around. Overactive forms of these proteins are often found in cancer cells. For instance, oncogenic RAS mutations are found in about 90% of the pancreatic cancers. RAS is a G protein, means that it switches back and forth between an active form and active form. Cancer-causing mutations often change RAS structures so that it can no longer switch to its inactive form, or can do so on only very slowly, leaving the protein stuck in the on state. In the presence of normal RAS, these proteins drive cell cycle progression only when growth factors are available, and the RAS remains on and the cell division and proliferation take place. And in the absence of the growth factor, the RAS remains off and there is no division and proliferation. And in the case of oncogenic RAS, even in the absence of growth factor, the cancer-causing mutations often change the RAS structure and it remains always in the on stage, and this results in excessive cell division and proliferation, resulting in cancer. Tumor suppressors. Negative regulators of the cell cycle may be less active in cancer cells. For instance, a protein that halts cell cycle progression in response to DNA damage may no longer sense damage or trigger a response. Genes that normally block cell cycle progression are known as tumor suppressors. Tumor suppressors prevent the formation of cancerous tumors when they are working correctly, and tumors may form when they mutate, so they no longer work. One of the most important tumor suppressors is tumor protein P53, which plays a key role in the cellular response to DNA damage. P53 acts primarily at G1 checkpoint, controlling G1S transition, which blocks cell cycle progression in response to damaged DNA and other unfavorable conditions. When a cell's DNA is damaged, a sensor protein activates P53, which halts the cell cycle at the G1 checkpoint by triggering production of a cell cycle inhibitor. This poses by time for DNA repair, which also depends on P53, whose second job is to activate the DNA repair enzymes as discussed earlier. If the damage is fixed, P53 will release the cell, allowing it to continue through the cell cycle. And if the damage is not fixable, P53 plays its third and final role, that is triggering apoptosis or programmed cell death, so that the damaged DNA is not passed on. In cancer cells, if the P53 is often missing or non-functional or less active than normal, For example, many cancerous tumors have a mutant form of p53 that can no longer bind DNA. Since p53 acts by binding to target genes and activating their transcription, the non-binding mutant protein is unable to do its job. Let's see again what's happening in normal p53 and non-functional p53. In normal p53, it detects the DNA damage and it acts as the G1 checkpoint, controlling the G1S transition, where it blocks the cell cycle progression in response to the damaged DNA. And this will halt the cell cycle at the G1 checkpoint by triggering the production of a cell cycle inhibitor. And also the DNA repair systems are activated, and if the repair is impossible means apoptosis or the programmed cell death will be triggered by the p53 so that the cells don't pass any damaged DNA. In the case of non-functional p53, the DNA damage is detected but the p53 cannot bind to the DNA. Here there is no halt at the G1 checkpoint and the cell cycle progression will continue. The DNA remains damaged and it is unfixed. So the damaged DNA mutation will be passed on to the daughter cells of such a division. So when p53 is defective, a cell with damaged DNA may proceed with cell division. The daughter cells of such a division are likely to inherit mutations due to the unrepaired DNA of the mother cell. Over generations, cells with faulty p53 tend to accumulate mutations, some of which may turn proto-oncogenes to oncogenes or inactivate other tumor suppressors. p53 is the gene most commonly mutated in human cancers, and cancer cells without p53 mutations likely inactivate p53 through other mechanisms. Example, increased activity of the proteins that cause p53 to be recycled. In animal cells, the extracellular growth factors control the progression of cells through the restriction point in late G1. In the absence of growth factors, cells are unable to pass the restriction point and become quiescent, frequently entering the resting state known as G0. When the growth factor stimulation occurs, they can re-enter the cell cycle. One critical link between growth factor signaling and cell cycle progression is provided by the D-type cyclins. Cyclin D1 synthesis is induced in response to growth factor stimulation, in part as a result of signaling through the RAS, RAF, MEK, ERK pathway, and cyclin D1 continues to be synthesized as long as growth factors are present. Cyclin D1 is also rapidly degraded if growth factors are removed. So cyclin D1 is a critical target of growth factor signaling. If any defect in cyclin D1 regulation cause the loss of growth regulation characteristic of cancer cells, for example, mutation resulting in continual unregulated expression of cyclin D1, contributes to the development of a variety of human cancers including lymphomas and breast cancers. Similarly, mutations that inactivate the INK4-CDK inhibitors are commonly found in human cancer cells, for example E2F and RD. You can see the induction of D-type cyclins here. The growth factors regulate the cell cycle progression through the G1 restriction point by inducing synthesis of D-type cyclins via the RAS, RAF, MEK and ERK signaling pathways. Retinoblastoma or RB was first identified as the product of a gene responsible for retinoblastoma, a rare inherited childhood eye tumor. RB is a prototype of a tumor suppressor gene, a gene whose inactivation leads to tumor development. The RB tumor suppressor protein play a key role in the regulation of the cell cycle. The RB gene product symbolized PRB. Early in the G1 phase of the cell cycle, PRB binds to the E2F proteins, a family of transcription factors that control the expression of several genes whose products move the cell through its cycle. When this binding occurs, they cannot bind to specific enhancer sequences in their target genes. Consequently, the cell cycle factors encoded by these genes are not produced, and the machinery for DNA synthesis and cell division remains quiescent, that is inactive or dormant. Later in G1, PRB is phosphorylated through the action of cyclin-dependent kinases. In this chain state, PRB releases the E2F transcription factors that have bound to it. These released transcription factors are then free to activate their target genes, which encode proteins that induce the cell to progress through S phase and into mitosis. In the first step, early in G1, PRB binds to the E2F family of transcription factors, and the bound E2F proteins are unable to stimulate transcription of their target genes. Later, the cyclin CDK complexes phosphorylate the PRB. Then the phosphorylated PRB releases the bound E2F proteins which activate their target genes. And the proteins encoded by the targets of the E2F transcription factors are involved in the cell cycle progression.